So, welcome to IPOPI webinar. Thank you all for joining this session. I am Martine Pergent, IPOPI president. I will be your moderator, and I would like to thank Sophie for making this webinar happen. Today, we are exploring the topic of hyperinflammation in PID patients and how this is different from hyperinflammation in COVID-19 patients. To address that theme, I would like to say a warm welcome to Professor Frédéric Rieux-Laucat from the National Institute of Health and Medical Research in CERN and the Imagine Institute in Paris. Welcome, Frédéric.
Thank you.
How will this webinar work? Professor Rieux-Laucat will soon begin his presentation. When he presents, you can post your question in the question tab that you see on the right side of your webinar screen. After the presentation, we will discuss a selection of questions with both Professor Rieux-Laucat and Dr. Nizar Malawi from Necker Hospital Paris and chair of IPOPI Medical Advisory Panel, who kindly has joined the webinar today. Hi, Nizar.
Hi, thank you, and welcome everyone.
He will also answer some of the questions, he meaning Nizar will also answer some of your questions in written form. And for that, we say thank you to you. Now, I will hand over to Professor Rieux-Laucat. Thank you for being with us today, and the floor is yours.
Thank you very much. So, good afternoon, ladies and gentlemen, and that's my pleasure to here share some results we obtained in the last months regarding the COVID disease. And in fact, if we were involved in that work, it's because we were working, as it was said, on patients with hyperinflammation due to genetic disease. And so the title that was given is Hyperinflammation in PID Explained and How It Differs from COVID-19. And I add a subtitle showing that how the study of rare diseases can help to understand the pathophysiology of severe forms of COVID-19. So here are the people in my team that were most of them at home due to the containment, but the people highlighted in red were volunteer to work and to collaborate on that topic. That I will show you the results in a minute. So if we talk about the immune responses to virus infection, in fact, there are two arms that can be depicted. On the right part of the slides, there is the protective immune response with regulated inflammation that will start with early response with inflammatory cytokines such as type 1 interferon, that will lead to the activation of monocytes and macrophages, pro-inflammatory cytokines and chemokines, and that will end up in a protective immunity, and then even with memory, with the interaction with the adaptive immune system. But then sometimes there is an inflammatory response that is leading to a pathogenic or dysregulated response, with a delayed type 1 interferon response, and then therefore robust virus replication, because the type 1 interferon is truly the first answer of the cell to a viral infection, and without a proper production of this cytokine, the virus will develop, stimulating then again the immune system, leading to increased and hyperinflammatory monocyte macrophages activation, as well as neutrophils, and pro-inflammatory cytokines and chemokines as well, that will trigger the infections or the invasions of the tissues by the monocytes, and that will lead, when it is in the lung, to acute respiratory disease syndrome, and sometimes, unfortunately, to death. So if we want to depict the cytokine storm we were hearing in the last weeks, there was the example of influenza virus that is very well known, where in fact after the virus infecting the lung epithelial cells, the first molecule at play will be the type 1 and type 3 interferon that will lead to the production of other inflammatory cytokines, and that is the first step, the innate immune response. That will be then followed in a couple of days or week to the adaptive immune response, and then the amplification again of this cytokine production and the production of antibodies, of course, and cytotoxic T cells. But the primary role is of interferons and IL-6, as well as other cytokines like the TNF, for instance. So that was very well known for a long time regarding the influenza virus infection. So talking about type 1 interferon, there are several receptors. So type 1 interferon has a single receptors, although there are 3 subtypes of interferon alpha. And there is also the interferon beta that is using exactly the same receptor on the left part of the scheme here. And the type 3 interferon is using another type of receptor, but they all go through the same signaling pathway using the JAK-STAT pathway after a cascade of phosphorylation. And therefore, the STAT, once phosphorylated, they heterodimerize or homodimerize and associate with other transcription factors like the IRF to then make the interferon-stimulating genes activation. And there is a second type of interferon that is called the type 2 interferon or gamma interferon that will also using the same pathway, leading to, again, the activation of hundreds of genes. So what can we measure? So when we, and that will be important for the follow-up on this presentation. So first, we can, of course, make the dosage of the production of type 1 interferon in the blood, in the plasma of the patients, using different methods. The ELISA that will have a sensitivity to nanograms or picograms per milliliter. But now we are using a more sophisticated way to measure that, and we use a digital ELISA, CIMOA, that can measure femtogram of the cytokine, so very, very sensitive. We can also measure the activity of the molecule. So the idea is that we put this, the blood of the patient, or let's say the plasma where the interferon is present, and we see how it can inhibit the VSV cytopathic effect in vitro, so that really not only measure the concentration, but also the activity of the molecule. Then following the pathway, we can measure the phosphorylation of the STAT molecules, and also then that will lead to the expression of hundreds of genes. Some have antiviral effectors function, some are negative regulators, some other are positive regulators. So we can pick some of these genes and measure them by quantitative PCR, and that will provide an interferon signaling gene score, measuring, in fact, the response of the cells to type 1 interferon. And that gives a good idea, and that shows that the cell was in contact with interferon and was stimulated by interferon. Regarding the IL-6, we can do more or less the same with the dosage in the blood, the assessment of the phosphorylation of STAT3 in the case of the IL-6 receptor that is depicted on the left here. And then we can also measure the expression of regulator genes, like the genes of the SOX family, by quantitative RT-PCR that will show that the cells were, again, stimulated by IL-6. Although for IL-6, the signaling is a little bit more complex because they are trans-signaling and the trans-presentation of the cytokines. I don't go into details, but basically we can make more or less the same dosage to assess the production and then the cell response to these cytokines that are key in the immune response to infections. So now I would like to summarize an example of a genetic predisposition to hyperinflammation responses and cytokine storm syndromes. And that was a rare disease that we described 6 years ago. in patients carrying activating STING1 mutation, and they develop vasculopathy and lung disease, and the acronym for this disease is the SAVI. So we described the first familial cases, so inherited mutations in 3 individuals. So they are heterozygous mutation, therefore they are dominant. And what we could see that these patients develop interstitial lung fibrosis, and that's when we had the opportunity to look in the biopsy of the lung of these patients, we could see that there was infiltrations of B cells marked by the CD20 marker, and the monocytes by the CD28 markers that were the main leukocytes that were infiltrating the lung of these patients. They also developed signs, cutaneous signs, and vasculopathy. In fact, STING is a DNA sensor. So STING is here in the cycle red. So in fact, the DNA sensor is cGAS. So when the cells gets infected by DNA virus or by bacteria, the DNA is recognized by this sensor that will produce a cyclic dinucleotide that will activate STING that is in the reticulum endoplasmic here. And these activations will lead to the translocation of STING through different compartments, and that will lead to the activation and phosphorylation of TBK1 and RF3 that will then induce the immune gen inductions of interferon, for instance. And therefore, when STING is activated, we can measure the type 1 interferon effect by the interferon stimulating genes expression. And as we could see at the steady state in the cells of these patients, in the T cell, the B cell, the monocytes, all cells in fact, there was an increase as compared to the control in gray of these interferon stimulated genes, showing that the cells are constantly stimulated by the type 1 interferon. These patients, they also produce uncontrolled, they have an uncontrolled production of inflammatory cytokines such as IL-6, here measured by the transcript, but can be also measured in the plasma, and of TNF-alpha in a less important way. So this genetic disease is in fact mimicking DNA virus infections, but it's a chronic, constant, and there was therefore this pathological cytokine storm, so to speak, because these increase and uncontrolled production of this inflammatory cytokine, starting with the type 1 interferon. So by identifying the pathway at play in this disease, so activation of STING, constant activation of STING, and therefore knowing the loop of activation, production of type 1 interferon, and then JAK STAT activation, and then again activation of these genes. There were drugs on the markets that were known to block specifically the JAK molecule, the JAK1 and the JAK2. So that was the rationale to treat these patients using these drugs, in order to block not the cause, but to block the consequences, and therefore to minimize the pathological consequences of this uncontrolled production of inflammatory cytokines. And in fact, so JAK1 and JAK2 inhibitors, ruxolitinib or baricitinib, they block not only the type 1 interferon here in the blue cycle, but they block also inflammatory cytokines such as IL-6 and interferon gamma. So we have a broad spectrum of cytokine receptors that could be blocked by these drugs. So since these drugs were not used in pediatric patients, we first started a compassionate trial that was supervised by Bénédicte Neven and Marie-Louise Frémont here in Necker to treat the first 3 patients. And that the results is here. So the fact that I don't have the, oops. I don't have the, I lost my, here it is. But you can see, so the lung, which is the most important here. So it is before treatment, and these patients were poorly responding to anti-inflammatory treatments. And after several months, you could see that there was a very good improvement of their lung status and their respiratory function, showing the efficacy of this treatment that is now given in a lot of patients that have this spontaneous production of type 1 interferon and inflammatory cytokine. So now, if we want to talk about immune therapies, there are different ways to block this inflammatory cytokine storm. And for instance, we can block the IL-6 or IL-6 receptors. There are a lot of monoclonal antibodies blocking this pathway. The anti-IL-1, as well as type 1 and type 2 interferon receptors, and then JAK inhibitors. But then, now in the context of the COVID, it could be a potential danger in inhibiting the immune response in the course of a viral infections, because, of course, these are dysregulated response to the SARS-CoV-2, for instance. Then if we block these pathways, are we at risk to acutize the disease, and therefore to be not beneficial to the patients? So that's why it was important to identify first the inflammatory cytokines that were at play in the SARS-CoV-2 infections in the context of the COVID-19. So when we compare the CT scan of the lung in the SAVI, so the genetic disease mimicking DNA virus infections, to the COVID-19 lung patients, we could see that they were very similar. So in the SAVI, we knew that there were high inflammatory markers, CRP and so on, and inflammatory cytokines. So the hypothesis that maybe in the most severe cases of COVID, it was also the type 1 interferon that was initiating a pathologic loop of cytokine storm, and that IL-6 may participate, and that may mediate the lung disease that sometimes runs the patient into a very bad shape, needing IC intensive care, and sometimes leading to death. So we started a project in collaboration with Benjamin Terrier and Solène Caneis at the CHU Cochin, the hospital just nearby, and with Nader Yatim and Darf Duffin, Pasteur Institute. And the question were the following: can we identify biomarkers of severity in adults patients with COVID-19? And these markers, could they be therapeutic targets? And is there a specific immune response in the pediatric patients correlating with the rare severe cases that we observe in the SARS-CoV-2 infections in children? I will focus my presentation on the first question, and therefore the primary objective was to rapidly collect within the first 2 months the detailed clinical and biological informations together with molecular and cellular data to better characterize the human immune response to SARS-CoV-2 infections, doing an immunological profile using a mass cytometry, sorry, high sensitivity plasmatic cytokine profiles using SIMOA to measure the production of these cytokines, the interferon type 1 response, and in the second step that is ongoing, to look at the single cell RNA-seq, so to look at the expression of all genes that are activated in response to the SARS-CoV-2 infections. So we defined a cohort of 50 patients, and that were classified as mild to moderate. So they had fever, cough, but no pneumonia, or mild pneumonia requiring low oxygenotherapy. Severe patients that are followed in the intensive care units that needed more than 3 liters per minute of oxygenotherapy, and those that were classified as critical, that required mechanical ventilation. So here are the informations regarding these patients. The median of age is around 50, so we excluded the patients, the old patients, because we know that they have that elderly is a comorbidity. Half of them have comorbidity factor, but there was no clear difference between the three groups of the patients, because it was very important to make the group of patients similar in order to drive conclusion on the analysis. So the first results here is showing the IL-6, the TNF, and the IL-1, and it is obvious here that going from the moderate to the more severe patients, there was an increase in IL-6 production, showing that there is a tremendous immune response at play in these patients. The TNF-alpha was also increased, whereas the IL-1 response was not increased in all patients. So higher IL-6 and TNF-alpha plasma levels in ICU patients were possibly a biomarker of severity, and therefore that may open the door to therapeutics like anti-IL-6 and anti-TNF treatment. I'm not going to detail that today. The surprise came that when we measured the type 1 interferon response in these COVID-19 patients, because first of all, the dosage of interferon alpha-2, for instance, but it's true for other, like interferon beta, that is undetectable, we saw that there was a reduced production of type 1 interferon in the blood of the most severe patients as compared to the moderate. That was the surprise, in contrast to our primary hypothesis. And that was even more obvious with the interferon antiviral activity measured in the plasma of the patient, that this activity, not in all, but in most patients, was profoundly impaired in the patient. When we put type 1 interferon ex vivo on the cells of the patients, we could see that they are all responding, and even the most critical, showing that the signaling pathway of the type 1 interferon receptor was not affected. So we have a lower type 1 interferon production, but a functional signaling pathway in ICU patients. And then we looked at the viral load in these patients, and whereas the viral loads in the nasopharyngeal biopsies or samples were the same in the different groups of patients, we could see that the viremia, so the viral particle in the blood of the patients, was increasing with the status of the patient. So the most severe patients having the most important viremia, and again, it's not universal. There are some patients that do not have viremia, but in most of them, they have high level of the virus in the blood. So the defective type 1 interferon production seems to correlate with a poorer control of viral replication in the most severe patients. Now, with all the immune phenotype analysis, I just show you some of them. So we saw that there was lymphopenia, total lymphopenia, that was more pronounced on the T cells and the CD8 T cells and NK cells in the most severe patients. And using markers that are showing that the cells are exhausted, so at the end of their life, so to speak, we could see that this exhaustion marker were more increased in the most severe patients, showing that there was an intense immune response following the SARS-CoV-2 infections, but that the most severe, those that are going to the intensive care units, have an exhausted adaptive immune response. Looking at the gene expression of the immune genes, so more than 500 genes, there is no need to be expert to see that. So when it's blue, it's low expression, and when it's red, it's high expression. So no need to be expert to see that the severe and critical patients have a high expression of some of these genes. And what was seen is that the NF-kappa B pathway was really at play, because going from no disease to moderate and severe and critical cases, we could see that the genes of this pathway were more expressed in the patient cells, that the NF-kappa B1 and NF-kappa B2, for instance, showing that there is an exacerbation of the activation of this pathway that is a natural immune response pathway, but that is even more importantly expressed in these patients. So the conclusion, so we starting from the hypothesis made on a rare disease, so patients carrying and activating STING mutation and producing a lot of cytokines that are making their lung disease and making this parallel between this rare disease and SARS-CoV-2 infections, we made the hypothesis that the cytokine storm should be sustained by high interferon, high IL-6 and TNF. But in fact, the most severe COVID-19 cases exhibited an impaired type 1 interferon production. And since this type 1 interferon is the most important cytokine to combat, to control a virus infection, that was leading to an uncontrolled viral replication. And in contrast to what was said in some cases, that the most severe cases were not dying from the virus, they in fact are dying from a viral infection, a very severe viral infections that trigger an exacerbated inflammatory immune response. So low type 1 interferon and high IL-6 and TNF are truly markers of severity, and that could potentially be a rationale for a therapeutic combination. And so there is in France a trial consortium that is called Co-Immuno, that is supervised by Olivier Hermine. They are trying different drugs, and they open a new arm in their drugs based on these results, where we should put, we could add in the patients interferon alpha in order to try to block the intense viral replication and therefore and then to block the cytokine storm, probably starting with anti-TNF, that is at the top of the cascade. TNF is activating the NF-kappa B pathway that will then turn into IL-6 production. But there could be also an arm of interferon alpha and anti-IL-6, for instance. What is the recommendation is that probably we should avoid the biologics affecting the type 1 interferon production. And one of the most efficient molecules to block the interferon production, type 1 interferon production, is the chloroquine. So, but I think the debate is closed now. So is it true for all severe cases? We don't think so, and we saw in some of the patients that for instance, the viral load was low, and that there was some good production of type 1 interferon. And it's all related to the host pathogen interaction during this SARS-CoV-2 infection. And here is a picture made from a review recently published by Mariam Merad, showing that what is known from the SARS-CoV-1, this epidemics that was in place 17 years ago. And it is known that the coronavirus may inhibit the pathway leading to the type 1 interferon production. In their genome, they have several molecules that can block at different level, and even blocking STING. So it could be that there are particularities in the genome of some virus in these patients, but it could be also that there is a particular genetic background leading to young patients or not too old patients without known comorbidities to develop a severe form when they are infected with the SARS-CoV-2 virus because of their maybe defective genes on this pathway. So I will stop here, and I would like to acknowledge the people that were involved in this work, so the people in my team. So now there is the second part of the project that is developed in collaboration with Michael Ménager, where we are going to look in details, cell by cell, which are the genes that are overexpressed or underexpressed. And as I said, that was a very nice and exciting collaboration with clinicians, Benjamin Terrier, Solène Kernéis, Nadia Yatim, and Daraf Delphine, the Institute Pasteur. I thank you very much for your attention. I would be happy to catch your questions. Thank you.
Thank you very much, Frédéric, for this great presentation. And this is certainly a very interesting topic and a tough one, but exciting one, as you said. Hello back to Nizar, because you're back there. And thank you for answering some questions, because we have many, in fact. Are there any PIDs that are more prone to be associated with hyperinflammation and autoimmune complications? or can it affect the patient no matter what the specific PID? This is the question. So are there any PID that are more prone to those dysregulation? And among them, is patients with STAT1 GOF mutation in great risk to severe COVID-19 infection due to higher risk to get hyperinflammation? So the generic one and about STAT1 GOF mutation.
Yes. So in fact, I'm speaking under the control of Nizar, but I think there were a few cases of PID patients that suffered from the SARS-CoV-2 infections. And so there was no dramatic cases. We had the example, for instance, of a patient with an LABA deficiency that was infected. His diabetes was dysregulated, but everything went. The evolution was very favorable. Then there are a lot of PIDs that are affecting the NF-kappa-B pathway and that are leading to inflammatory disease. In addition of the one that I was talking, so SAVI, but also the interferonopathies, so the Aicardi-Gouttière syndromes, for instance, they all are producing a lot of type 1 interferon. But it's also true in patients that are carrying mutations of NF-kappa-B1, A20. But since these patients, I would say that if their PIDs is not affecting the production of type 1 interferon, which is clearly, and other studies over the world are going in the same way. So type 1 interferon is the key response in front of virus infection, and in particular coronavirus. So if their immune system is able to make and all their cells type 1 interferon in the right time and in the good amounts, they should not suffer so much from these coronavirus infections. And they will not probably, because they are always regulations, although they have inflammatory disease that is not completely regulated, but they will not develop this probably hyperinflammatory response, because if you keep in fact the take-home message that if you keep the replication of the virus down, then the immune system will be regulated by itself, and then once the virus is controlled, the response goes down. You cannot exclude that in some cases where you have a dysregulation of these NF-kappa-B pathways, IL-6, IL-1, you may see some inflammation, but usually these patients are also under anti-inflammatory drugs, so that may control their disease. So I think there is a balance, and I won't worry too much about these patients regarding the SARS-CoV-2 infection.
Mm-hmm.
Yeah, I totally agree with you. Just to be very quick, all kinds of primary immune deficiencies can be associated with auto-inflammatory or inflammatory or autoimmune features, CVID, for example, or CGD, or Wiskott-Aldrich. But of course, there are some specific PIDs that are more prone to develop such complications, such as the IPEX, for example, or the ALPS. But that's for the general, PID population. And for the with regards to the COVID-19, as Frédéric mentioned, as far as we know, all patients, PID patients who we know got this infection, there was no representation, higher frequency of PID patients belonging to the groups that are more prone to develop autoinflammatory or autoimmune diseases.
And along to this theme, is the cytokine storm described in COVID-19 a manifestation that also can be found in PIDs?
Yes, there are PIDs that are known to, in fact, have a cytokine storm and following infections, and they are the PIDs where there is a defective cytotoxic functions of the T cells. So therefore, these T cells are no longer able or less able to kill the infected cells, and they get stimulated constantly, and then they have these cytokine storms, so there are the perforin diseases and all related diseases. And it's true that these patients, when they get infected by a herpes virus, for instance, like EBV, they start to develop the macrophage activating syndrome that is related to the cytokine storm, with IL-6 again in the middle, and interferon type 2, interferon gamma this time, are the key molecules. But they are, yes, there are similar cytokine storm in PIDs, in specific PIDs.
Okay, and maybe another maybe tough question, I don't know. Do you think that immunization with BCG could be protective to COVID-19?
Yes, it's a tough one.
Not sure.
Well, there are some papers showing that by stimulating the immune system, you may help the response against the coronavirus. But I would say that if you don't have, again, the first response to the infection is the type 1 interferon. If you don't produce at the right time, in the right time window, and in a sufficient amount the type 1 interferon, then you get into trouble. If you get this interferon, so therefore, if you vaccinate with the BCG, you will stimulate this innate immune response first. But regarding the adaptive response, so antibodies and T cell response, that they will not be specific for the virus. So now there is a notion that people that have encountered other coronavirus, the one that we usually get infected by during the winter, they may have cross-reacting antibodies, and therefore if you stimulate the whole system, you may think you may favor a response, but I would not consider that this is the standard of care for the COVID infection.
Yeah, another question, Nizar, if you want to just pop in. Thank you. Another question, some clinicians using cytokines blockade in moderate patients before reaching ICU status. Do you agree with that?
Sorry?
Some clinicians use cytokines blockade in moderate patients before reaching ICU status. Do you agree with that? Yes.
I mean, if we can assess so if we can guide the decisions through the assessment of the cytokine dosage, that may provide a good reason to block the cytokine storm before. Then, of course, it has to be used with cautious, because if you block IL-6, for instance, that is also a very important cytokine in response to different infections. You may develop other co-infections. So this has to be regarded and very cautiously and monitored with all other possible co-infection events. And then, of course, these patients, they are in the hospital, they are in specialist centers, so they are monitored. But that could prevent the patients to go to the intensive care units. Mm-hmm. But again, we have to be very, very cautious with these treatments. We should not forget that in most cases, the immune system and the cytokine production is making the job. So if patients that goes to the hospital for pneumonia, I think without comorbidities or risk really to develop the severe form, it could be a bit too much to block these cytokines. That's what we are doing with the RNA-6 single cell analysis. We are trying to identify markers that would predict that some patients are at risk to develop the severe form, and that would guide the clinicians to use these biologics or not.
Yeah, I will raise two last questions. The first one is that true that there is a different profile of COVID-19 patient reaction, maybe one driven by IFNI and other creatine activation syndrome, maybe more driven by IL-1, and in what proportion?
Well, I have absolutely no idea of the last part of the question. As I said, what we found is not universal. There are some of the severe patients that made good interferon, so it may well be that they have a problem with DRL1 or DRL6. We know that there are some papers about to be published using the anti-IL-1 antibodies or the anakinra, yes, and with good results. But again, I think the problem is that it's not. I think that no one can say that he has a universal treatment or a magic potion, you know, like a druid, and it may differ from patient to patient, and but then by assessing some easy parameters like production of type 1 interferon, IL-6, IL-1, TNF, measuring the viral load may already help to decide and to decide which drug we can use. But today we can say that the trials are not finished, but all these different type of treatments, anti-IL-1, anti-IL-6, anti-C5a are already under trials. The fact is that it was difficult sometimes to include the patients because someone claimed that using antimalarial drugs could be useful, but therefore the patients wanted these treatments. But in fact, these trials are still ongoing, and the science needs time, and it is necessary to measure the effect of these trials with a group with the treatment and a group without the treatment to be sure that it is effective. But of course, yes, it may vary from patient to patient, and I would certainly not say that giving interferon alpha and anti-IL-6 would be the magic question.
Okay, and the last question would be maybe more prospective, but do you think your research work on COVID-19 hyperinflammation mechanism could lead to new treatment pathway for PID patients as well? And eventually elaborate a little bit on that, even if theoretical.
Well, I think that new drugs are not going to be used in the COVID, because they are, in fact, what they are doing, they are trying to repurpose the existing drug, and most of them are already used in the PID patients. But we can understand some mechanisms, and for instance, when we are working with these patients with high interferon production, sometimes we don't know whether they have a concomitant infection. So we will learn how to address this inflammatory response to see whether it's an early response due to infections, whether it's a chronic response due to a genetic defect, and that will help us in better understanding the pathophysiological mechanisms in PIDs, certainly.
Thank you very much. Is there any comment?
No, it's the best conclusion for this brilliant webinar. Thank you very much, Frédéric.
Yeah, so thank you both. We come now to the end of this webinar, which was made possible thanks to a kind donation from Sobi. On behalf of IPOPI, I would once again like to say thank you to Professor Rieux-Laucat for your participation, and also to Dr. Malawi for his contribution. Thank you again to all of you for attending this IPOPI webinar. You were close to 200, and this made us very happy. Please note that you will be able to see the recording on IPOPI TV within a couple of days. We encourage you to share it as much as you can to those you think will benefit from it. Take care, and see you soon with another webinar. Bye-bye.